Hey everybody, in this video we're going to take a look at the nucleus and division of the nucleus or mitosis. You've probably heard at some point in high school biology or something that the nucleus is the brain of the cell or the control center or the computer of the cell. That's definitely a metaphor. It is a library, maybe would be a better metaphor, that stores all of our genetic material, but it is at least partially responsible for how that genetic material is expressed. So to that extent, it is a bit like our cell's computer, but really it's a storage. It's a DNA storage center. Not every cell even has a nucleus. Red blood cells, for example, do not have a nucleus, and for that reason they don't live very long, 3 or 4 months, and they can't divide, but they are a nucleate. Other cells have more than one. They could have a lot more than one. Skeletal muscle cells, for example. Osteoclasts, I believe, can be multinucleate. Those are bone-destroying cells, and some liver cells can be multinucleate. Typically, if a cell has multiple nuclei, it's a pretty big cell. It has a lot of cytoplasm, and it has a lot of demand for proteins and enzymes and things to be made by the nucleus. But most cells just have one. There are 3 distinguishable regions of the nucleus. The first is the nuclear envelope. It's a lot like the cell membrane, except that it is actually 2 walls with a fluid cavity in between those 2 walls. So it's more similar to the mitochondrial membrane. There are protein complexes that form pores that span this membrane that allow certain things to move in and out of the nucleus, like messenger RNA, for example. And within that nucleus is the nucleoplasm, which contains chromatin and our nucleoli. So our nucleoli are dark staining regions within the nucleus that produce ribosomal subunits. So that is actually where ribosomes are made. At least their subunits are made here, and then they leave the nucleus and they're assembled out in the cytoplasm. Some of them just float around in the cytoplasm. Those are free-floating ribosomes. Some of them bind to the rough endoplasmic reticulum and produce protein there. Those are membrane-bound ribosomes. Those subunits are made in the nucleoli. And then lastly, we have our chromatin. Our chromatin is our genetic material. It's 30% DNA, 60% histone protein. Histone proteins are, they're like little discs, very small, obviously, and they arrange in stacks of 2. So if you can picture 4 stacks of 2, almost like, I picture like a Spree candy or like a Mentos candy. If you picture 4 stacks of 2 of those right next to each other, the DNA actually wraps itself around twice those 8 histone proteins. So when you have 8 histone proteins and 2 strands of DNA, or 2 wraps of DNA around those 8 histone proteins, that's actually called a nucleosome. And a nucleosome is that fundamental unit of our genetic material, which is called chromatin. So anyway, back to our percentages. So it's 30% DNA, 60% histone, and 10% RNA. And again, that DNA wrapped around the histone protein is called nucleosomes. And then we have millions and millions of those nucleosomes. And when we zoom way out and we look at it in the nucleus, it just looks like this jelly-like substance called chromatin. And that's perfect. It's unpacked. It's easy to access. It's easy to go in there and read a gene and produce a protein based on that gene. So chromatin is perfect for that. Chromatin is perfect for the day-to-day need for genes to be expressed. What it's not very good at is when it comes time to divide. So when it's time for the nucleus to divide, which is called mitosis, chromatin is not really the best form for our genetic material to take because it's a fluid, and it's impossible to cut a fluid in half and have the exact right genes on one side and the exact right genes on the other. It's just not organized. So we have another form of genetic material called chromosomes. We only see chromosomes during mitosis. So when it's time for the nucleus to divide, the chromatin will condense and become chromosomes. Chromosomes are these little X's here in this picture. So this bar on the left, which is called a chromatid, it's one of the sister chromatids, is identical to this bar on the right. It's sister chromatid. So we have two sister chromatids that are identical. They're joined in place here in the middle by something called a centromere. When it comes time to divide the nucleus, all we have to do is split that centromere, and we can take one identical copy of DNA, put it into one nuclei, and the other chromatid goes into another nuclei, and now we have two identical sets of DNA, and we do that for our 23 chromosomes. This diagram here is showing the cell cycle. There's two main phases. One is called interphase, and one is called M-phase. So back, I'm not sure exactly when, but back a long time ago when we were figuring this stuff out, scientists would look at a cell and they'd see a whole lot of nothing happening for a while, and they'd call that interphase. Nothing happening, nothing happening. And then once in a while, or pretty frequently, depending on the cell type, they'd see this crazy thing happen where the nucleus would split in half, and then the cell would split in half, and then we'd have two new cells. They called that M phase. And then they decided, well, M phase is exciting, so we'll give it a name, and then we'll call the whole rest of the cell cycle interphase because nothing much is happening. We now know that quite a lot is happening in interphase, depending on the cell. Mitosis is just when it divides. So anyway, those are the two phases of the cell cycle, interphase and M phase, division and the time between division. You may hear interphase called the metabolic or growth phase. It's probably a better term for it. The cell is growing. It's performing all types of important functions during interphase. And it has subphases. G1 subphase, not a whole lot of preparation for division is occurring here in this first, maybe third of interphase. The centrioles start dividing during this phase. If the cell never divides, like a neuron, we say it's in G0 phase. But G1 is just a growth phase, a metabolic phase. Centrioles are dividing. S phase is a very important phase. This is when we're copying our DNA. The S is for synthesis. So this is a synthesis phase when we're duplicating our DNA so we can divide it in half. During G2, we're making all sorts of important proteins and enzymes necessary for mitosis. The centrioles have finished dividing, and we'll talk more about that in mitosis. And we are now, at the end of G2, we are now ready for mitosis. And then that will be the next slide. We're going to cover DNA replication in another video. We may actually not get to it, but we will cover that in another video. It deserves its own video. It's pretty interesting. And we'll also do protein transcription and translation in another video potentially. Let's get into mitosis because that's really the point of this particular video. So, mitosis is the division of a nucleus. It's often referred to as a division of a cell. Mitosis is technically division of a nucleus. It's typically accompanied by cytokinesis, which is a division of a cell, but it doesn't have to be. Mitosis is specifically nucleus division. It's also called M phase. It has 4 phases within it with some key events that we will discuss in each phase. Prophase, metaphase, anaphase, and telophase, or PMAT, P-M-A-T, is a good way to remember that. We're going to look at each phase and a couple key events of each phase. All right, so prophase is our first phase, and we split it up into 2 subphases, early and late prophase. In early prophase, the chromatin condenses to form chromosomes. Remember, that's important so that we can divide the chromosomes equally. The centrosome, which if you remember, is this structure with 2 centrioles that the microtubules radiate out from. It's divided already during G2 phase. It's split in 2. There's 2 of them. During early prophase, a spindle forms between them and the centrosomes start to separate. You can see the spindle. These are microtubules pushing against each other, pushing the centrosomes to opposite poles of the cell. So chromatin condenses, forms chromosomes. Centrosomes separate, mitotic spindle forms. That's early prophase. In late prophase, the nuclear envelope dissolves, so now these chromosomes are just floating out in the cytoplasm. Spindle microtubules attach to kinetochores at each centromere, and polar microtubules slide past each other, forcing the poles apart. Okay, let's back up for a second. There's a lot of words there that we don't recognize. So, unfortunately, everything seems to start with the C and rhyme with chromosome or chromatid or centromere or centrosome. So let's just do a quick recap of all these words. The centrosome refers to this yellow structure here with the two centrioles. There's two of them now in mitosis, and throughout the phases of mitosis, we'll see that they're pushing apart to try to split the cell in two. Meanwhile, they have a structure called a kinetochore microtubule. Not all of the microtubules in the mitotic spindle are pushing apart. Some of them are attaching to the centromere, which is the center of the chromosome that holds the two sister chromatids together. There's a protein in that centrosome called kinetochore, and these microtubules attach to that kinetochore protein and they start to pull on the chromosome. So not only are the centrosomes pushing the poles of the cell apart, but the kinetochore microtubules are playing tug-of-war over the chromosomes. And we'll see the result of that in the next couple slides. So recap, late prophase, the nuclear envelope dissolves. The spindle microtubules attach to kinetochores at each centromere. Polar microtubules slide past each other, forcing the poles apart. So spindle microtubules, sometimes I'll call them kinetochore microtubules. Those are playing tug-of-war over the chromosomes. They're attached to the centromere, specifically the kinetochore at each centromere. And the polar microtubules are pushing the poles of the cells apart. In metaphase, wow, what did I write there? I think I was just correcting centrosome, not centromere. So during metaphase, the centrosomes are at opposite poles of the cell. So here they are on each side. Centrosomes are opposite poles of the cell. They got there because the polar microtubules are pushing the spindle apart. The chromosomes are also aligned along the equator or metaphase plate, which you can see here I drew in blue. They all got here because the spindle microtubules that are attached to the kinetochore at each centromere are pulling, and they're pulling equally, so the chromosomes align along the metaphase plate. And now, during this phase, enzymatic separation of the chromatids begins. So let me erase this really quick. This is a chromosome, okay? It has two sister chromatids, 1 and 2. The sister chromatids are held together by something called a centromere. That centromere has two kinetochore proteins, and there's a microtubule attached to each, and is pulling on it. And then what we just learned is that enzymatic separation of those chromatids is beginning. So this little yellow centromere is beginning to be dissolved in this phase. Now, if you can imagine, we have spindle microtubules tugging on these chromosomes, and we're starting to degrade this little centromere. Eventually it's going to split. And that is exactly what happens during anaphase. During anaphase, the sister chromatids separate. Once they do, now we stop calling them sister chromatids, and we start calling them chromosomes again. So this is a chromosome, and this is a chromosome. They're daughter chromosomes instead of sister chromatids now. The motor proteins within the kinetochores pull the microtubules towards the poles. That's why they look like little V's, because they're being dragged. They're actually moving, so they look like little V's. The polar microtubules continue to expand, pushing the poles farther and farther apart. Which brings us to our last phase, telophase. Telophase starts when the chromosomes stop moving. It looks a lot like prophase in reverse. The chromosomes revert to chromatin. A new nuclear envelope forms, nucleoli reform, the mitotic spindle degrades and disappears, and mitosis ends. The cell is now binucleate. The cell has two nuclei that are identical. Cytokinesis is the division of the cell into two new cells, each with an identical nucleus. An actin ring forms around the cell and constricts until the cell is pinched in two. This begun during anaphase, and it will continue past telophase. And guys, that is it. That is the nucleus, mitosis, and cellular division called cytokinesis. Thanks.